All right, good work. We are over halfway through with this section. Hopefully at this point you are finished copying the figure, exploring the cell cycle into your notes. Turn to the next page, please. This is the start of section 3, the final of the 3 stages of cytokinesis. Go ahead and write this section heading into your notes, and then you can pause the video and read the section. The first thing that I see in this section, indeed it's in the first line, is a vocab term. Cytokinesis is the final stage of the cell cycle. But it's more than that. Notice that the bold words underneath that definition give us even more information about what happens during cytokinesis. I'm going to add that to my definition just so it doesn't get accidentally lost as I'm using this text or using my notes to study for a quiz or to take my to study for a test or to take my open note quiz. During cytokinesis, the cytoplasm divides distributing organelles into each of the 2 new cells. So not only do the cells divide, but all those organelles inside the cell are split up as well. Now we know that an animal cell is different from a plant cell. We talked about that in chapter 1. Because of these differences, they have to divide in slightly different ways. So in an animal cell, I'm going to write this in a different font just so that my eye is drawn to it. The cell membrane make this not a down. There we go. Squeeze this together around the middle of the cell. The cytoplasm, so inside the cell membrane, that jelly-like substance, the cytoplasm, is going to pinch in 2. Again, here we go with half of the organelles in each daughter cell. Half on one side, half on the other. Now conversely, in plant cells, a cell plate is going to form. And as this is happening, that's an and sign. You could do a plus sign or you could write A and D. It's totally up to you. A new membrane is going to develop. That is what allows for the 2 new cells to completely detach. You have that cell plate, which is sort of like a new cell wall, and then the new cell membrane dividing or developing on either side. The last thing I notice in this section that's important is that at the end of all of this separation of the cells, each daughter cell has to have the same number of chromosomes as the original parent. We don't have new daughter cells that are different. We have new daughter cells that are exactly the same. When you're finished with that, go ahead and read the next section called the length of the cell cycle. When you're done, unpause the video and we will continue. This is a short section, and I believe we could probably summarize it in one bullet point. Let's see, the length of each cell cycle, we'll copy right out of the book, varies from cell to cell. Now the visual that the book gives us is also sort of interesting to show how, in this case, this is the average human liver cell, the amount of time in interphase is way longer than the amount of time that is actually spent in mitosis or cytokinesis. Gives us an idea of how these stages are broken apart. You don't have to copy that into your notes today, but it's an interesting thing to check out. It gives you some additional perspective on what's happening. The next section, if you turn the page, is called DNA replication. Go ahead and read that through. When you're done, unpause the video. Don't forget your section heading. All right, there are a few important things in this section. The first is really obvious. The authors wanted us to catch it. They put it in bold. So let's see, DNA replication ensures that each daughter cell will have all the genetic information it needs to carry on its activities. Let's summarize that. It's okay if you don't summarize in the same way that I do. We all think slightly differently. The key is to not take too many shortcuts in our summarizing. We have to make sure that what we're writing accurately reflects the ideas that are there, that the author has written down in the textbook. So it's okay to try to cut some words out. Just don't cut out so many words that when you go back to use your notes later, you don't know what you were talking about. So cells copy DNA before mitosis begins. There we go. To ensure that each daughter cell has genetic information needed to carry out its activities. Now there's some interesting information about pioneer scientists in the 1950s. The first part is sort of a general factoid, but then it gets to be more specific in that those last two sentences. We learned that in 1953, anytime we get a specific date, focused information, it's probably good to include. In 1953, two scientists, one named James Watson and the other, remember that we can use plus sign and sign, however we want to say and, another called Francis Crick, figured out the structure of DNA. Now this is really important. They actually won a Nobel Prize for this discovery, I believe, because knowing how DNA is shaped helped us to understand how it works, how it replicates, what all it's able to do. That is the end of the section on DNA replication. Go ahead and turn the page and read the section called replication process. Oops. I think I skipped something, didn't I? The structure of DNA. Go ahead and read the purple-headed section, the structure of DNA. When you're done, we will pick up. The first thing that I learned when I was reading this section was that the shape of the DNA molecule has a name. The DNA molecule is called a double helix. Now the book goes on to explain what a helix is, and if you aren't sure, this would be an awesome piece of extra information to have in your notes. So a helix is a shape that twists like threads on a screw. Here's an example of a visual that is perhaps helpful if you don't really get the visual picture from just the definition of a shape that twists. Having the comparison to it being like a thread on a screw can be helpful. Now it goes on to describe the DNA as being sort of like a ladder. The DNA is like a ladder that is made up of two different types of molecules. The first is called deoxyribose. Now you might remember from class that I've used that term to describe DNA. We said that the name for DNA, if we were to spell it all out, is deoxyribonucleic acid. Deoxyribose, that sugar that's a part of DNA, is the D in the acronym that we're so used to seeing. So deoxyribose sugar is part of the equation. The other part is a phosphate molecule. In between each of these ladder pieces, we have rungs. So like the places where you put your feet on a ladder. Each rung on the DNA ladder is made up of a nitrogen base. That's what the book says. If this isn't all completely making sense right now, do not fear. Remember that we will talk about all of these notes and do some activities with them during class. The notes are designed to get you into the content. This isn't your only opportunity to see it or your only opportunity to learn it. Now the next thing I see in the book is another list. Remember, whenever I see a list, I'm going to want to try to put that in a numeric list form. So there are 4 kinds of bases. The first one is called adenine. And then if you look later on, it says that they're abbreviated in different ways. And adenine is abbreviated with the capital letter A. So I'm going to add that in right now as well. The next one is called thymine, and that's abbreviated with the capital letter T. Then we have guanine. That's kind of a fun one to say. It's abbreviated with the capital letter G. And then last but not least, on a separate page here, sorry folks, is cytosine. And that is abbreviated with a capital letter C. The book goes on to explain that these 4 kinds of bases can only pair up in very particular ways. The A, the adenine, is only going to partner up with, and you see a W and a slash, that's my way of writing out with, it's only going to pair up with T, the thymine. Likewise, G, guanine, is only going to pair up with C, cytosine. I want to make sure I understand that, so I'm going to put that into my notes. Now, once you're finished, you can go ahead and turn the page, and we have our very last section called the replication process. Take a moment, please, to read that section through. Pause the video. When you're done, push play, and we will continue. All right. Now I'm sure, because I've been trying to type and talk and work with screens all at the same time, that I have made a number of typos. Some of them are probably pretty funny. If you see typos, I apologize. Feel free to bring them to my attention later on, but unfortunately I'm not going to be able to go back and edit individual parts of the video, so bear with me on those, please. The last section here we have is called the replication process, and there were a number of interesting things in this section. I noticed that it's set up, well, to describe a process, and a process usually has steps. So I'm going to go out on a limb, and I'm going to assume that I can find a way, if this is truly a process with steps, to write this section out as one giant list. So in the DNA replication process, what are the steps that are going to happen? Well, the first thing I see is that 2 sides of the DNA molecule are going to unwind and separate. That's good to know. The second step in this process appears to be that the nitrogen bases, those 4 kinds of bases we talked about up here, are going to pair up. So there were nitrogen bases floating around, and they're going to start to pair up with each of these halves that have unwinded and separated. The third thing I see is that when new bases are attached, this is sort of wrapping it up and putting a nice bow on it, 2 new molecules are formed. And that is the end of the replication process. So we were able to take that section and break it down into 3 simple stages. Your book wants you to understand that just like at the beginning of the section when we were saying that the daughter cells are going to be exact replicas of their parents, these new bases that have formed out of the DNA strands that unwound and separated are going to exactly match the original DNA molecules that started the process. So let's put that in. My book really wants us to understand that in this section, it appears. They keep saying it. And that, I think, is a great place to end this section.